Welcome to the cell-cell communication during development module. In this talk, we're going to learn about cadherins. We'll first learn about the role of cadherins in development and describe the quantitative effects of cadherin interaction and the outcomes on cell movement and sorting during development. We will also learn about the qualitative effects of cadherin interactions on the cell movement and sorting during development. Morphogenesis is the process of arranging cells in such a way that they create organized structures and shapes. This process is very important for the development of organs and allowing these organs to function. The main types of cells that are present during development are epithelial and mesenchymal cells. The epithelial cells are able to adhere to one another, and so they're able to form complex structures like sheets and tubes. Mesenchymal cells, on the other hand, are loosely connected to one another, and more than often they are not connected to one another at all, which allows them to migrate. They're able to migrate to different places and are able to lay down the extracellular matrix. Now, organs are usually formed from epithelium and the underlying mesenchyme. During development, the cells have to have a way to interact and communicate with one another so that they can arrange themselves spatially and temporally in such a way that productive organs are formed as development progresses.
So now we're going to look at the basics of cell-cell communication. Now, cell-cell communication is mediated by proteins of a cell that can be secreted into the surrounding environment, or these proteins can be present on the cell membrane, or they can be inside a cell. Now, communication between cells can be local or they can be long range. And in this chapter, we're going to look at two specific types of cell communication, which are the juxtacrine types of communication, which results in juxtacrine signaling, and the paracrine types of communication, which result in paracrine signaling. So when we look at the figure to the right, and when we look at the figure that's part of the box, we're looking at juxtacrine signaling. And this is normally mediated by cell surface proteins that are present on two cells that interact with one another. When the same type of protein from one cell interacts with the same type of protein in another cell, it is called as homophilic binding, which is what we can see with the two yellow-colored membrane proteins on both cells. However, if you have one type of protein from one cell interacting with another type of membrane protein on another cell, then what we are seeing is called as heterophilic binding. During juxtacrine signaling, you can also have interactions between the cell and the extracellular matrix. Paracrine signaling, on the other hand, uses secreted molecules that can be called as paracrine factors. These are usually secreted from one cell, and these paracrine factors are sensed by another cell through the presence of cell surface receptors. We're going to see more details about juxtacrine and paracrine signaling as we go along the chapter. We're now going to look at a hypothesis called as the differential adhesion hypothesis. And what this hypothesis basically was built on the fact that during development, cells do not sort randomly, but actively move to create tissue organization. The embryologist Steinberg performed experiments that showed that certain cells migrate centrally when combined with certain cell types, but migrate peripherally when combined with other cell types. Now, cells rearrange themselves into the most thermodynamically stable pattern. And hence, this migration usually depends on the strength of adhesion between the cells. The proteins that mediate adhesion are different types, but the one that we're going to look at that play important roles in development are the cadherins. Cadherins are calcium-dependent adhesion molecules that are membrane proteins. they're able to bind to cadherins on other cells through their extracellular domains. So when we look at the picture to the right, here we can see the extracellular domains of the cadherins present on two different cells binding to each other and forming interactions with each other. On the cytoplasmic end, the cadherins can interact with actin through the proteins called cadherins. So in the figure, when we look at the upper part of the figure, we can see that at the cytoplasmic side, the cadherins are bound to these proteins called catenins, and the catenins associate with actin. Thereby, cadherins are able to interact with the actin cytoskeleton inside the cell with the help of the catenin molecules. Cadherins play a very important role in the developing embryo, and different types of cadherins are expressed by different types of cells. Let's look at some examples of cadherins that are present. So E-cadherin is a cadherin that's usually present in all cells of the embryo during early embryogenesis and plays important roles during the events that occur during early embryogenesis, especially during gastrulation. P-cadherin is a cadherin that helps the placenta stick to the uterus. N-cadherin is another type of cadherin that helps in nervous system development, while R-cadherin helps in retina formation. Another type of cadherins are the protocadherins that help in keeping migrating cells expressing the same class of protocadherins together. Thus, the different cadherin molecules can actually help in the developmental process of different organs. The strength of cadherin interactions plays an important role in determining cell surface tension and the tendency of cells to bind together. The strength of the cadherin interactions depend on the quantity or how many cadherin molecules are present on the cell surface, and it also depends on the qualitative interactions between the cadherin molecules, which includes the kind of cadherins that are expressed and when these cadherins are expressed. When we look at quantitative effects of cadherin interactions, we see that cells are able to sort themselves based on the amount of cadherin that is actually expressed. So cells with more cadherin expression tend to migrate internally. When we look at the figure, here we're actually seeing two different cell types, green cells and red cells. Now the green cells have more cadherin expressed than the red cells, which have less cadherin expressed. And as we can see with time, the green cells that have more cadherin like to migrate internally and remain in the inside, and they are normally surrounded by the red cells that did not have that much cadherin expression. So in this case, we can say that the green cells had stronger adhesion and hence migrated internally, while the red cells did not have strong adhesions or had weak adhesions, and that's why they remained at the periphery. Another example of the quantitative effects of cadherin interactions is that in many instances the cell sorting hierarchy is strictly dependent on the amount of cadherin interactions between the cells. So when we look at this picture, let's look at the first panel where we have cells that are expressing P-cadherin or E-cadherin. P-cadherin is expressed more than E-cadherin, then in that scenario, the P-cadherin cells are going to be present internally while the E-cadherin cells will be at the periphery. If we have equal interactions between P-cadherin and E-cadherin, then we do not see the migration of cells and they are uniformly distributed. However, if the P-cadherin cells, which are in red, are lessly expressed than the E-cadherin cells that are in green, then the E-cadherin cells tend to be internal while the P-cadherin cells are on the periphery. Thus, depending on the expression of how many cadherin molecules are there, cell migrations can be determined. In addition to the amount of cadherin expression, the type and timing of cadherin expression is also crucial for development. The type of cadherin plays an important role in cell sorting, and we're going to look at an example where we have two populations of cells expressing either R cadherin, that's going to be shown in red, or B cadherin, that's going to be shown in green, and they are present at equal levels and are mixed together. Now, if we look at the figure here, what we can see is these two types of cells sort out into opposing mounds of cells with a distinct border between them. Thus, the R cadherins expressing cells like to stay together, and that's where we see the red mound. And the B cadherin expressing cells, which are shown in green, they like to stay together. And so these are the qualitative effects, or the type of cadherin that is determining where the cells are going to be migrating. Cadherin expression must also be timed properly for correct development. When we look at the initial stages of embryonic cells, initially all the embryonic cells express E cadherin, but the cells that have to become the neural tube will lose E cadherin and gain another type of adherin or start expressing the cadherin called N cadherin. If the expression of N cadherin is altered, the neural tube development and nervous system development is hampered due to a lack of proper cell migration. So in this figure, when we look at the portion that's in the blue box, we're looking at the development in a wild-type embryo where it expresses the normal form of N cadherin. And in this case, when we look at the second type of picture, which is towards the right, in this case, we're looking at an embryo that does not express N cadherin. Now let's focus at the red boxes. When we look at the wild type, we're able to see that the cells have moved in certain ways to form that V-shaped kind of structure. However, when we have the N cadherin mutant cells, we don't see the cell migration happening, and hence proper border bind formation has not occurred. This border formation is important for nervous system development. Thus, the type of cadherin and the timing plays a really important role in development.
With this, we come to the end of our talk where we learned about cadherins and the quantitative and qualitative effects of cadherin interactions on cell movement and sorting during development.